Hi students, this video will explain about gaseous exchange and its control, subtopic 7.1 gaseous exchange and control in mammals. The learning outcome is for us to able to describe the structure of hemoglobin and its characteristics as respiratory pigments. Then you need to able to describe three ways of carbon dioxide transport from respiring tissues to lungs. Then analyze the oxygen dissociation curve of hemoglobin. Then compare oxygen dissociation curve of hemoglobin and myoglobin. And the last one, E, is to analyze the effect of changes in partial pressure of carbon dioxide towards oxygen dissociation curve. What is gaseous exchange? Gaseous exchange is a transfer of gases between an organism, for example human, and the external environment in either direction. For example, inhalation, gases from atmosphere into our lung, and exhalation, gases from our lung expelled out into the atmosphere. So the transfer of gases occurs by diffusion across a concentration gradient, meaning that we need to understand, like we have learned before about the simple diffusion that involve movement of substances from high concentration to low concentration. So it's across the concentration gradient and includes the exchange of oxygen and carbon dioxide by the alveoli of the lungs. So in this topic we are focusing on the exchange of oxygen and carbon dioxide. The air tract to alveoli from atmosphere, it passes through our nasal and buccal cavity, then to pharynx, trachea, bronchi, bronchioles, and the last one, the subunit, is the alveoli. Alveoli is a very thin layer of tissue cells. It's a one cell thick in our lung. Let's look at the characteristics of erythrocytes, how they are adapted, or what makes them a very efficient oxygen transporter. First, erythrocyte has biconcave shape. If you were to explain why having a biconcave shape is good for erythrocyte as oxygen transporter, because having biconcave shape will help to increase the surface area for gas diffusion. Do not just stop at increase the surface area. So you have to relate with the increase of surface area for gas diffusion. Erythrocyte has no nucleus, so having no nucleus, it provides more space for hemoglobin. For example, biconcave, let's say that it has a nucleus here. So the polypeptide would have a limited or less space because it has been filled by a nucleus. Then erythrocyte has the ability to generate ATP by anaerobic respiration. Okay, why this is good? Because anaerobic respiration only produces 2 ATPs, right? But how does this ability give benefits? The benefit is for us because erythrocyte will not consume the oxygen that they transport for their own use. If they were to undergo the aerobic respiration, then they have to use the oxygen and produce more ATP. But instead, erythrocyte will only generate ATP by means of anaerobic respiration, so that they won't use the oxygen that they transport. Let's has a recap of the structure of hemoglobin. You have learned about hemoglobin in semester 1, in first semester, in Molecular of Life, when you learn about the protein. So hemoglobin is a conjugated protein because it is attached with a non-protein molecule, which is called prosthetic group, or specifically the heme group. So it is a conjugated protein made up of a globular protein. A globular protein means they have a 3D structure. The polypeptide are packed and twisted and folded into a globular protein. Since hemoglobin has 4 subunits of polypeptide chain, so they are grouped as quaternary level structure of protein. Okay, it's a quaternary level because it has more than one polypeptide. Okay, the polypeptide are alpha chain and beta chain. Each subunit, each polypeptide contain one heme group. So for each hemoglobin, they have 4 heme group, and for each heme group, they has ferrous ion, one ferrous ion, which is capable of carrying one oxygen molecule. So for one hemoglobin, they will be able to carry 4 oxygen molecules. Since one molecule of hemoglobin able to carry 4 oxygen molecules, if we're looking at the fact, one molecule of erythrocyte consists of or carries 250 million hemoglobin. 250 times 10 power of 6. So if we times by 4, because each hemoglobin able to carry 4 oxygen molecules, then we have 10 power of 9 oxygen molecules for each erythrocyte. Now let's look at the hemoglobin. Why they are respiratory pigment? First, hemoglobin has high affinity for oxygen, meaning that they can bind oxygen even at low partial pressure of oxygen. So now we are going to use the terms partial pressure. We won't use concentration of oxygen, even though partial pressure is the concentration, partial pressure of oxygen is exponential, directly exponential towards concentration of the oxygen, meaning the higher the concentration, the higher the partial pressure. So hemoglobin has high affinity for oxygen, meaning that they can bind oxygen at low partial pressure of oxygen. So the binding of oxygen to the hemoglobin will form oxyhemoglobin. The oxygen will bind to the ferrous ion, the heme group. So in the lungs, we are considering the oxygen, the hemoglobin are saturated with oxygen in the lungs, because in the lungs the partial pressure of oxygen is high. When the hemoglobin reach an area or tissue that has low partial pressure of oxygen, it will easily release oxygen. So this characteristic is very important because it will ensure that the tissue that is respiring, that needs the oxygen, will receive the oxygen that is carried by the hemoglobin. Okay, so when oxyhemoglobin arrive at the respiring tissue, it will tend to dissociate, forming oxygen and hemoglobin. So looking at this equation, we can see hemoglobin binds to oxygen. Oxygen binds to hemoglobin. This occurs in the alveoli, forming oxyhemoglobin. When the oxyhemoglobin arrive or reach tissues, respiring tissues, the partial pressure of oxygen is low, so oxyhemoglobin will dissociate and release oxygen. So oxygen will diffuse into the tissue. Hemoglobin has cooperativity characteristics because one hemoglobin consists of 4 subunits. So if all of the 4 subunits does not cooperate, they might be able to only carry 1 or 2 or 3 oxygen. Not all of the hemoglobin will be cooperating to carry the oxygen. So cooperativity is a characteristic that increase their affinity. So it means that the binding of oxygen to the first subunit will induce the remaining subunit. We have 4, so the first subunit binds to oxygen. The rest of the subunit, the 3 other subunits, will change their conformation and increase affinity for them to bind to oxygen, another 3 oxygen. So this results in oxyhemoglobin, meaning the hemoglobin, all of the parts of the hemoglobin, 4 polypeptide, will be bound to the oxygen, forming oxyhemoglobin. If not, then the oxyhemoglobin is not completed. Okay? The fourth characteristic is about their binding, the binding of the hemoglobin, ability of the hemoglobin to bind to the oxygen loosely and reversibly, meaning that it is not permanent and the binding is reversible because it goes back to the second characteristics. The hemoglobin can easily release the oxygen. Okay. The fourth characteristics, part 2, is the ability of the hemoglobin to bind to another gas, which is carbon dioxide. So, the binding of carbon dioxide to hemoglobin is at the amino group, not the heme group. It is not attached to the heme group. Instead, it is attached to the amino group of hemoglobin. Remember, hemoglobin is a protein. So protein subunit is the amino acid. Do you still remember the structure of your amino acid? C double bond OH. This is the carboxyl terminal or carboxyl group. The R, the H. And then we have the amino terminal, the amino group. So carbon dioxide bind to this part of the protein, forming carbaminohemoglobin. That's why we do not name the binding of carbon dioxide, the combination of carbon dioxide to the hemoglobin as carboxyhemoglobin, because it does not bind to the heme group. So it binds to the amino group. It is called carbaminohemoglobin. And the fourth characteristic, sub 3, is the ability of the hemoglobin to bind to another type of gas, which is carbon monoxide. What's bad about binding of hemoglobin to carbon monoxide is carbon monoxide bind to the hemoglobin at the heme group, forming carboxyhemoglobin. Thus, it is considered as a competitive inhibitor to the oxygen. And furthermore, the binding of carbon monoxide to hemoglobin is irreversible. So this causes the ability of the hemoglobin to bind to oxygen becomes lower. Thus, it lowers the affinity for the hemoglobin to bind to oxygen. Not lowering, lowers. And this causes an interruption to gas exchange, because it prevents oxygen from binding to the hemoglobin. The exchange of gases occurs between the blood and the lung. Lung being the alveoli, specifically, and the blood itself. The blood is in the blood capillary, and blood capillary are surrounding the lung, the alveoli. Yeah? The atmospheric oxygen transferred from lung to bloodstream. Okay, let's say this is the alveoli. This is the chamber where the oxygen comes from the atmosphere, and this is the blood capillary, and we have the erythrocyte in the blood capillary. So the oxygen partial pressure of oxygen is high in the alveoli, so it will diffuse into the blood capillary. The partial pressure of oxygen in the blood capillary is low. Okay, and so oxygen gas will diffuse from alveoli into the blood capillary, and it will bind to the hemoglobin in the erythrocyte, while the blood is carrying carbon dioxide, so the partial pressure of carbon dioxide is high in the blood capillary and low in the alveoli. So carbon dioxide will diffuse out of blood capillary into the alveoli. The exchange of gases depends on the partial pressure of the gases in lung and blood. What is partial pressure? Partial pressure is the pressure of one type of gas that is proportional to its concentration. As the concentration increases, the partial pressure will increase because it's in the mixture of gases and not affected by other gases. The transport of oxygen. The partial pressure of oxygen in alveoli is high. So you have to remember this. Partial pressure of oxygen is high in the alveoli. So oxygen will bind to hemoglobin, forming oxyhemoglobin. Okay. In the lung, in the alveoli, because the alveoli are surrounded by blood capillary. So the red blood cell in the blood capillary, this is the alveolar space. So oxygen will diffuse from alveoli into the erythrocyte, forming oxyhemoglobin. While in respiring tissue, the partial pressure of oxygen is low because the oxygen is being used by the tissue itself. So the oxyhemoglobin will have the tendency to dissociate and release oxygen. So oxygen will diffuse from erythrocyte into the respiring tissue. How does the oxygen diffuse from alveoli into the blood capillary? What makes them move easily from alveoli into the blood capillary? Because the partial pressure in alveoli is high, while partial pressure of oxygen in the blood capillary is low. So oxygen will diffuse from alveoli into blood capillary, then bind to hemoglobin, forming oxyhemoglobin. Then the oxyhemoglobin will be transported to the tissue throughout body. Okay, when the respiring tissue has low partial pressure of oxygen, so the blood capillary will have higher partial pressure of oxygen. That's why the oxyhemoglobin will dissociate and release oxygen. Oxygen will diffuse out of the blood capillary into the respiring tissue. Okay, now let's explain the transport of carbon dioxide. Carbon dioxide in the respiring tissue, a partial pressure of carbon dioxide in respiring tissue is high due to the respiration, because respiration releases carbon dioxide through Krebs cycle and link reaction. So the carbon dioxide from the mitochondria will move to the cytoplasm, then diffuses out of the cell into the capillary blood capillary. Then carbon dioxide has a characteristic that is they are soluble in water. So because blood has H2O, has water, so it will the presence of the carbon dioxide will be affected. Okay, will be affected because it can dissolve in water. So we are going to explain about how carbon dioxide are transported from tissues to the respiratory surfaces, the alveoli. So to carry carbon dioxide, okay, let's say the carbon dioxide is diffused from the respiring tissue into the blood capillary. Okay, what happened to the carbon dioxide once it crosses to the blood capillary? 10%, some of the carbon dioxide, 10%. Let's say we have 100 molecules of carbon dioxide. 10 of them will dissolve directly in the blood plasma because our blood plasma has water. Okay, another 90% will enter the RBC. Okay, out of 90 molecule of carbon dioxide, 30% of the 100 will bind to the erythrocyte, the hemoglobin, to form carbaminohemoglobin. While 60%, you have to memorize the percentage, 60% of the blood will enter the RBC and then transported in the form of bicarbonate. Okay, we will explain this in details. Because along with this one, because the formation of when the carbon dioxide dissolve in the blood plasma and the erythrocyte, another further reaction occurs. Okay, in respiring tissues, it has higher partial pressure of carbon dioxide and low partial pressure of oxygen. So carbon dioxide will diffuse out of the respiring tissue into the blood capillary. So in the blood capillary, carbon dioxide will combine with H2O, forming carbonic acid. So the formation of carbonic acid is catalyzed by carbonic anhydrase. Okay, carbonic acid, the formation of carbonic acid in the blood plasma occurs slowly. Then carbonic acid will dissociate into hydrogen ion and bicarbonate ion. So it will stay in the blood plasma in the form of bicarbonate ion. What happened in alveoli? Because when they reach alveoli, the bicarbonate ion will reversely bind with the hydrogen ions and form carbonic acid, and then carbonic acid dissociate into carbon dioxide and water. So carbon dioxide will be released into the atmosphere along with H2O. That's why when we breathe out, when we exhale, we have water vapor together in our breath. Okay, as carbaminohemoglobin, the carbon dioxide combine with amino group of the hemoglobin to form carbaminohemoglobin. When they reach the alveoli, the carbaminohemoglobin dissociate to form hemoglobin and carbon dioxide. So this is carbaminohemoglobin. Okay, the 60%. This is what we are going to explain. The explanation is more. Because carbon dioxide from tissue diffuse into plasma then enter erythrocyte. Yang 10% tadi, carbon dioxide diffuse into the plasma and form H2CO3. So the same thing actually occurs, but it occurs in the erythrocyte. Okay, forming by carbonic acid, catalyzed by carbonic anhydrase. So because it is catalyzed by carbonic anhydrase, an enzyme, so this reaction occurs faster. Then carbonic acid dissociate into hydrogen ion and bicarbonate ion. So the same thing occurs, but this occurs in the erythrocyte. Okay, I remind you, all of this occurs in the erythrocyte. What happens next? The bicarbonate ion that formed in the erythrocyte diffuse out of erythrocyte and then transported in blood plasma into the alveoli. So bicarbonate ion will be transported in the blood plasma even though it is formed in the erythrocyte. Because bicarbonate ion diffuse out of erythrocyte, it makes the erythrocyte becomes more positively charged because what's left is the hydrogen ion. So to make it balance, because we don't want the positively charged erythrocyte, so we have to counter the positive charge by having chloride shift. So what is chloride shift? It occurs when the chloride ion from the blood plasma, the salt that we consume. So this is one of the benefits. The chloride ion will diffuse into the erythrocyte. So to to replace the bicarbonate ion that diffused out of the erythrocyte earlier. So if you were asked to give the importance, the function, and the significance of chloride shift, it is to maintain electrochemical neutrality, because positive plus negative should become neutral. So the presence of chloride ion does when they enter the erythrocyte, they do not bind to any molecules. So they are just there to maintain the neutrality of the erythrocyte. Okay, let's not forget about the hydrogen ion. The increase of hydrogen ion in the erythrocyte would cause the erythrocyte to become more acidic. Okay, more acidic. So to prevent the hydrogen ion to lowers the pH, it needs to be held by something. It needs to be held by something. We cannot expel the positive ion out of the erythrocyte just for it to for the sake to make the erythrocyte less acidic or more electrochemical neutral. But what we can do is we have the hemoglobin as the buffer. Buffer maksudnya penimbal. So the acidity should be buff. Maksudnya kita timbalkan balik supaya dia tidak menjadi keasidan tak meningkat. Okay, what does the hemoglobin do is it will form a bond, or it will bind with the hydrogen ion, forming hemoglobinic acid. Okay, how? First, it has to dissociate the oxyhemoglobin. So this is oxyhemoglobin. That's why it will release or dissociate by releasing the oxygen. So oxygen will move out of the erythrocyte. Hemoglobin now is available, so it will bind to the H+. So once the hemoglobin bind to the H+, it will prevent decrease in pH, prevent increase in acidity. Okay, the 60% of the carbon dioxide that is transported in RBC as bicarbonate ion carried in the blood plasma, then arrive at the alveoli. So what happened next is the bicarbonate ion diffuse back into the erythrocyte. Masuk balik dalam RBC. So the hemoglobinic acid just now will dissociate. So the hydrogen ion released from the hemoglobinic acid will combine with the bicarbonate ion, forming carbonic acid. Okay, the enzyme carbonic anhydrase will proceed catalyzing. Will proceed the conversion of carbonic acid to carbon dioxide and H2O. So it can do reversible work, carbonic anhydrase. Then carbon dioxide diffuse out of the erythrocyte and moves to alveolar space. So this is the diagram that shows a summary of what I've explained before. The first step is the first method is the 10% carbon dioxide dissolved in plasma. Next, we have 30% of the carbon dioxide diffused into the erythrocyte and combined with hemoglobin to form carbaminohemoglobin. And the third one, the 60% of the carbon dioxide enter the erythrocyte, but then diffuse out of the erythrocyte in the form of bicarbonate ion. So this is what happened in alveoli, the reverse action. So we can separate this one. This is what happened in the alveoli, in the lung. And this is what happened in the tissue. Okay, loading and unloading of oxygen. Oxygen only be carried in the form of oxyhemoglobin by hemoglobin. So the hemoglobin involved in loading and unloading of oxygen. It depends on the cooperation among the hemoglobin. So this is the cooperativity characteristics. One oxygen bound, one hemoglobin, one hemoglobin, one hemoglobin, one hemoglobin. So it will induce the rest of the hemoglobin to bind to oxygen. That's why it is considered as increased affinity. Release of oxygen from one subunit will cause the other three subunits to release the oxygen along. Factors that influence loading and unloading. What is the indication? What is indicator that tells the hemoglobin to release the oxygen, to unload the oxygen? First, the partial pressure of oxygen. When the partial pressure of oxygen is high, the hemoglobin will have the tendency to load oxygen. So this usually occurs in the alveoli, the area that has high partial pressure of oxygen. While when the partial pressure of oxygen is low, more oxyhemoglobin will dissociate, then oxygen is unloaded. So this usually occurs in the tissue. The second factor is the partial pressure of carbon dioxide. When partial pressure of carbon dioxide is low, the hemoglobin will have the indication that they need to load the oxygen, forming oxyhemoglobin. For example, in alveoli, low partial pressure of carbon dioxide, and then in tissue, when the partial pressure of carbon dioxide is high. So when partial pressure of carbon dioxide is high, more oxygen will dissociate from oxyhemoglobin. So it tells, when the partial pressure of carbon dioxide is high, it tells the hemoglobin that the area needs more oxygen. Then the hemoglobin needs to carry the carry out the carbon dioxide to the lung. The third factor is pH. When the pH is high, it is basic lah. So pH is high, more oxygen bind to Hb, meaning when pH is high, less carbon dioxide. Okay, so just remember, high pH will increase the tendency of hemoglobin to bind to oxygen. High pH. When the pH is low, oxygen will tend to dissociate from oxyhemoglobin. So pH low, oxyhemoglobin dissociate. So the loading and unloading, due to the different partial pressure, different pH, produce oxygen dissociation curve for hemoglobin. The oxygen dissociation curve of hemoglobin shows a relationship between percentage of hemoglobin saturated with oxygen. Okay, how many of the hemoglobin is saturated with oxygen? It means that is the meaning of it. How many hemoglobin is saturated with oxygen? Okay, let's look at this. No hemoglobin is saturated with oxygen. So kalau sebab hemoglobin ada 4 subunit. Kalau satu sahaja, 1/4 sahaja yang mengikat dengan oksigen, jadi dia belum tepu lagi lah. So it needs to be bound with all of 4 subunits need to be bound to the oxygen, then it can be considered as saturated. Even though only, that's why here 0 ni doesn't mean that they are not bound to the oxygen, but it means that there is no hemoglobin that is fully saturated with the oxygen. Maybe only one hemoglobin only 25% oxygen. 1/4 sama dengan 25%. Jadi maybe only one oxygen bind to one hemoglobin. Okay, so as the partial pressure of oxygen increase, it shows an increase in the numbers of hemoglobin that is saturated with oxygen. So if we have like 100 hemoglobin, okay, it might be 70% here at partial pressure of 40 of oxygen, partial pressure of oxygen 40 mmHg. There are 70% hemoglobin are saturated with oxygen, meaning out of 100 there will be 70 hemoglobin that is saturated with oxygen. 70 times 4, because each hemoglobin saturated with oxygen, meaning all of 4 subunits are bound with oxygen. Understand? The sigmoid shape, this is called sigmoid due to cooperativity of hemoglobins. What can we say when what happened at here and what happened at here? At low partial pressure of oxygen, the oxygen has to struggle to bind with hemoglobin. That's why kat sini kosong, kat sini hanya ada dalam 10%. So kalau ada 100 hemoglobin, hanya ada 10 hemoglobin sahaja yang penuh dengan oksigen. Yang lain-lain mungkin baru 1/4, 2/4. Okay? Sebab tu there is sudden increase here because of the cooperativity. Okay? The binding of oxygen to one subunit here, sebab tu dia naik ni exponent, induce the remaining subunit to change their conformation and increasing the affinity of hemoglobin towards oxygen. So more the hemoglobin, more of the subunit will bind to oxygen. Okay. One hemoglobin starts to become saturated with oxygen. Ni dah dah penuh. Ada yang dah penuh, ada yang belum. Okay. Jadi kalau kat sini area ni ada yang dah penuh, ada yang belum lah. Okay. When partial pressure of oxygen is low, as in respiring tissue, percentage of oxygen saturated with hemoglobin is reduced. So at respiring tissue, low partial pressure of oxygen, usually around here. When partial pressure of oxygen is high, usually here. Percentage of oxygen saturated with hemoglobin increase. Oxygen combined with hemoglobin to form. Now we are going to explain the oxygen dissociation curve of hemoglobin and myoglobin. But before we continue with the explanation, we need to remember the fact that hemoglobin has a quaternary level because it has more than one polypeptide chain. It has 4 polypeptide chain. While myoglobin is a tertiary level protein because myoglobin has only one polypeptide. Okay. Myoglobin, for the ODC, myoglobin has a different shape of ODC compared to hemoglobin. Myoglobin has a hyperbolic shape while hemoglobin has sigmoid shape. What is myoglobin? Myoglobin is a red pigment in the mammalian muscle. So it is not in the blood capillary. It is in the muscle. It doesn't move. It consists of single polypeptide chain with one heme group. So the function of myoglobin is to store oxygen in skeletal muscle. It is not for oxygen transport. When myoglobin combines with oxygen, it will form oxymyoglobin. Myoglobin shows higher affinity for oxygen compared to hemoglobin. This will cause the oxygen dissociation curve for myoglobin shifted to the left. Meaning that, okay, this is the graph. ODC for hemoglobin is sigmoid, while for myoglobin it is at the left side of the ODC for hemoglobin. Okay, so this is for the myoglobin, this is for the hemoglobin. So it is said to be shifted to the left and having hyperbolic shape. Oxymyoglobin, the fact is oxymyoglobin will only release oxygen when the partial pressure of oxygen in skeletal muscle is very, very low. This is suitable for their function as oxygen storage. Okay, now how to calculate amount of oxygen released from tissue? Okay, the maximum saturation is considered as 98%. Okay, at 98%. Then we minus the maximum minus with the percent of saturation of oxygen at 20 kPa. Okay, ni sebab soalan dia 20 kPa. So we go high. So at 20 kPa, what is the percentage of hemoglobin saturated with oxygen? So this is how many of the oxygen is released. Then that's why we have to minus 98 with 20. Then we get the 78%. Why 98%? Because the rest of the 2% is dissolved in the plasma. We are considering that not all of the oxygen are bound to the myoglobin. The hemoglobin. Okay, now let's look at the Bohr effect, still under oxygen dissociation curve, but we have something that we call as Bohr effect. Bohr effect is due to the increase in partial pressure of carbon dioxide. Okay, so what is Bohr effect? Bohr effect is about the displacement of ODC. Kalau bahasa Melayu kita panggil anjakan, anjakan. Anjakan ni yang kita sebut tadi shifted to the left or right, shifted to the left or right. So for Bohr effect, it shifted to the right biasanya. Due to the increase of partial pressure of carbon dioxide, because the increase of carbon dioxide related to the decrease in pH, acidity. So once the carbon dioxide, if the carbon dioxide, partial pressure of carbon dioxide increase, the pH will decrease. And this is usually associated with a respiring tissue. So at respiring tissue, the ODC of hemoglobin is shifted to the right. Okay, the effect, Bohr effect ni, is very important. Kenapa bila ada Bohr effect ni important? Sebab bila hemoglobin, the ODC of hemoglobin shifted to the right, meaning it shows that the oxyhemoglobin is unloading or releasing the oxygen. So it is to ensure more efficient oxygen delivery from oxyhemoglobin in respiring tissues. The unloading. Remember just now I've already explained the unloading of oxygen. Oxyhemoglobin dissociate. What makes the oxyhemoglobin dissociate? Because the high partial pressure of carbon dioxide. So Bohr effect is important to ensure that the oxyhemoglobin will dissociate and release the oxygen. So Bohr effect ni is the displacement of ODC of hemoglobin. So yang displacement tu, inilah dia beza dia. Okay, at normal pH, at 20 partial pressure of oxygen, we will have 35% of hemoglobin saturated with oxygen. But if, when we do exercise, our carbon dioxide release is increased because our tissues are respiring more. So the pH of the blood will decrease. So when it decreases, it will drop the percentage of the hemoglobin that is saturated with oxygen. So it will release, it shows that it will release 10% of the oxygen is released to ensure that the respiring tissue can get more oxygen. Okay, can have oxygen during the respiration. So increase oxygen unloading. So yang shift to the right ni lah dia, because at lower pH, the curve will dia dah duduk, kita tak kata dia ke bawah, kita kata dia ke kanan. Walaupun kita nampak sebenarnya dia nak merendah kan. It lowers, it shows a decrease. Tapi we say shifted to the right. Shifted to the right maksudnya bukannya ni bermula di sini, tak. Mula tempat yang sama tapi duduk di bawah garisan yang normal tadi. So in alveoli, partial pressure of carbon dioxide decrease, pH increase. Okay, when pH increase, it is an indicator that the hemoglobin needs to load the oxygen. That's why at pH, higher pH, the ODC is at the left side. When it's at the left side, it shows hemoglobin has higher affinity towards oxygen. So meaning that more hemoglobin are formed, more oxyhemoglobin are formed. While in respiring tissue, The cellular respiration, the respiring tissue release more carbon dioxide, increasing the partial pressure of carbon dioxide. So carbon dioxide reacts with water, forming carbonic acid, and then release H+. So this is the cause of the decrease in pH. So decrease in pH will decrease the affinity of hemoglobin for oxygen. It tends to dissociate or unload the oxygen. So that's why ODC of hemoglobin shift to the right. Okay, this is when the tissues are respiring more. Ni example in alveoli. So ni macam kita compare pH alveoli dengan pH respiring tissue, and at the same time kita akan nampak perbezaan oxygen saturation of hemoglobin dengan tisu dengan yang berlaku dekat tisu. So dekat tisu lebih sedikit oksigen yang diikat kepada hemoglobin sebab hemoglobin tends to dissociate. Okay. Thank you for 7.1. We will meet at 7.2 later.